Hello, everyone. Welcome to Reporting at Room Now, and I'm hoping you're following the ACR 2021 Convergence meeting. We'd like to talk on some important topics around TNF inhibition. So, Ariel, do you want to tell me who you are and then what you'd like to discuss today?
Hey, sure, absolutely. So I'm Dr. Aurelie Najm. I'm from Glasgow. Today, the few abstracts I want to discuss are about drug antibody monitoring, little n. I know David has to tell about that as well. So I'm going to make the good introduction for him. Then I also would like to talk a little bit about all the different disease domains in PSA and how TNF inhibitors can act. And finally, I'll talk a little bit about about single cell monitoring, single cell analysis, pathway analysis, and how maybe it can help, direct treatment in the future. So just very briefly to go back to the disease domains. So it was abstract 1818. It was in the Correvitas registry, and it's a prospective registry of PSA and spondyloarthritis patients above 600, follow-up 7 years on TNF inhibitors. And what they did was actually cool because they looked into TNF inhibitors maintenance and persistency over time, and also duration to stop based on the different domains.
I couldn't hear what you said.
Oh, actually my Siri got activated by my voice. All right. Anyway, so they looked into the different domains, peripheral arthritis, skin disease, enthesitis, dactylitis, and so the persistency of the drug was actually about 75% over 6 months and dropped to 39% over 24 months, which is basically pretty much basic figures that we know for TNF inhibitors in PSA. But see, interestingly, there was no difference in persistency based on every single domain. The only thing that they showed was that actually the time to stop was shorter if people had in the domain of enthesitis or actual PSA, which we also know because these are the most difficult to treat. But also it was longest with patients with oligoarthritis. Now, moving on to heterogeneity in PSA and single cell analysis and translational research, the abstract 1816, which was using the Select PSA1 study, patients' samples. So just for memory, you probably all know it. It was comparing upadacitinib, adalimumab, and placebo in about 300 patients. And what they did is that they took sera from these patients and they did single cell protein biomarker analysis using the Olink technology. This technology allows you to analyze loads of biomarkers and proteins at the same time, and they did pathway analysis. And the thing that they showed that was quite interesting, I thought, was that upadacitinib was more targeting adaptive immunity, and especially T cells and B lymphocytes, and also the pathways with bone resorption and angiogenesis, while adalimumab was more likely to act on innate immunity pathways. I think this is quite interesting because this could start unraveling different disease mechanisms or different treatment mechanisms, and this could be a basis for personalized therapy. Now, personalized therapy can also be about drug monitoring, and this is where I'm going to talk about number 1525, which was actually looking at the randomized control trial Nordrum A, which, as you probably know, was negative. There was about 400 patients, and they look into infliximab immunogenicity and anti-drug antibodies. And they were just reporting that the factors that were associated with positivity of these anti-drug antibodies, first of all, 19% of the patients overall had antibodies and associated with smoking and a longer drug holidays. So more than 11 weeks basically was just predictive of having anti-drug antibodies, while a higher dosage of infliximab or an increase in dosage or other immunosuppressive treatment were reducing that risk. And interestingly, they showed that RA was predictive of having anti-drug antibodies. I think it's just because RA patients just get lower dosage. I mean, at least that could be an option, but that was pretty much it for me.
Right. And they probably, RA probably makes more antibodies, hard to say. Yeah. So let me get it straight. You're telling me that persistence of TNF inhibitors in psoriatic arthritis is, first of all, it's not great. We're not doing as well as we'd like. By a couple of years, it's way down, and they followed for 7 years, and you've lost like more than 2/3 of the cohort at the end. You're saying that single cell biomarkers show a difference between UPA, basically looking at a JAK kinase inhibitor, and adalimumab looking at a TNF, but yet on head-to-head, they did kind of about the same. And then you're telling me that anti-drug antibody monitoring that maybe would be a solution for some of the TNF inhibitors, at least in this trial, really didn't help us very much. So it kind of seems like we had a triple header of not very positive things, although not that negative either. So David, I guess, do you have a different spin on drug monitoring?
Well, there is slightly better news on the horizon. And I think I'm very grateful to Aurelie for setting that up for me, because I think it does come down to, I think there's been a lot of negativity in rheumatology about therapeutic drug monitoring in the past because we haven't been able to show it works. But Najm, so firstly, I mean, I'll say that it's obviously very philanthropic of the Norwegians to do these kind of studies, because they can do this in a well-organized way, in a centralized way, without necessarily concern for money. You know, this is a kind of liberty that oil buys you, right? And the difference between Najm A and Najm B is that Najm B is looking at proactive TDM rather than reactive TDM. And I guess taking what Aurelie said about the antibodies, I mean, you'd want to think that once you start on this cascade of necessity and developing anti-drug antibodies, it's hard to catch up on that. You're really chasing something which is very hard to try and overwhelm. And our IBD colleagues certainly have more success in overwhelming it than we do. But what if we were to get to the point where, instead of the randomness that we were talking about in previous days of essentially decreasing or increasing something like infliximab dose just on a whim, what if we were to do it on the basis of drug levels? And that's what they did in Najm B. So really what they've done, they had a set strategy based on drug levels. So basically, if your drug levels were in the range, keep them there. If they were low, increase it up. If they were very low, then try and potentially, and if there were anti-drug antibodies, switch away. And then if they were very high, potentially reduce them down, and essentially guide yourself on that basis. And on that basis, and they looked at this over multiple different diseases. In fact, it washed out that it actually favored therapeutic drug monitoring across all diseases. But then when they looked at subcategories, including spondyloarthritis, what they've classified as spondyloarthritis there, then there was benefit as well. And in fact, if you look at time to disease worsening, and there's some really nice Kaplan-Meier This presentation has only just happened a few minutes ago before this panel. We saw that essentially the hazard ratio with this type of approach was actually 2 compared to, the hazard ratio of standard therapy worsening is 2, so basically twice as the TDM approach is twice as good. So really in that situation, I think that gives us a lot of optimism. Now, I think we need to drill down on the details. We don't know at this point in time what the standard approach was in terms of what people did in terms of reduction. Reductions were allowed there. This was an open label study, so people could easily have been taken down different pathway and believed that this more kind of scientific approach was actually better. But at the same time, it's the first time we've seen TDM work in rheumatology for TNF inhibitors, and I think that's a big moment.
It's a big moment, but I think we have to not hold our breath because of the fact that therapeutic drug monitoring has been negative in at least 2 IBD trials, even though there's still a belief system. But I mean, it makes sense. It would be like antibiotic levels. It would be similar to when we're monitoring INRs on a drug like warfarin, that it would make sense to try to get people into that target range. However, number needed to treat to improve, one person is unknown right now, and number needed to harm with side effects. You know, there is a decent therapeutic window on a drug like infliximab, adalimumab, et cetera, but there's also cost to getting one benefit. So I think it's pretty exciting. It kind of makes sense. And when we already just heard that retention is not as great as we would like on any of our drugs, including TNF inhibitors and drugs like, or in diseases like psoriatic arthritis, why not do the best we can do? So I'm for it. It would be great to have some of these drugs flat dose pricing. That would be unbelievable. So with the Clin Farm hat on, I think you're going to say more will come. Let's see what happens.
More will come. Let's see what happens.
You got it. I think I'll say that too. Moving along. So what about looking at drug comparativeness on basically safety? So Eric, I think you were going to tell us a bit about skin cancers.
Yeah, I think from a TNF perspective, this was something that interested me a lot because this is part of the conversation you have when you start TNF. I always talk to patients about seeing a dermatologist and increased risk of non-melanomatous skin cancers, so basal cell and squamous cell cancers. So I was very interested to see abstract 1942, which was the rabbit cohort from Germany, and they presented their data on risk of non-melanomatous skin cancers on various biologic medications. And what was a little surprising to me was really the one thing that stood out was abatacept above all the other biologic medications. So TNF and the other medicines really didn't show as much of a signal as I had expected to see. And what they saw was a hazard ratio of about 2 for abatacept. I think there's a couple things within that analysis that this is a cohort study, and the patients that were on TNF inhibitors, they were more likely to be first line or earlier biologic use. Some of the patients on abatacept were further along in their disease duration, and it may have had other risk factors and other things as well. But I think certainly skin cancer is something that we have a lot of concern with abatacept for particularly melanoma. I think that's something to have in the back of your mind. I think there's a little bit more information that I'd want from a TNF picture there. The other thing that was also noted was that patients with co-medication with methotrexate may also have more of an increased signal. And again, was this a sicker population? Was there something else? Is something that I think will be interesting for more information. But I saw overall kind of reassuring from the TNF picture, but I think it's definitely something to watch with abatacept a bit further.
Right. So I think what you're telling me is TNF performed better than we thought, but the abatacept were further along, more comorbidity, maybe sicker, and that kind of confounding or channeling bias might sort of damn a drug when maybe the risk is the same amongst groups. I think time will tell, but it was a nice presentation, a large N, and I think it's just something to keep in mind with all our patients. And there are data from Australia where methotrexate has increased skin cancer in various diseases, not just our rheumatic diseases, but there are other studies, a US registry where they looked at hydroxychloroquine and methotrexate, where the skin cancer risks were the same. And so it's really tough to know it's increased or it's not. It depends on probably the risk factors and where your ozone layer, where you live, ozone layer or not. And I think going back to Glasgow, there's not a ton of sun there. So skin cancer there would be less, although people are fairer, perhaps in Scandinavia and Glasgow and Celtic kind of genes, than maybe moving along to other areas where sunburns might increase your skin cancer risk too, and all the usual confounders. So with that in mind and looking at the differences, Swetha, I think you have something to tell us about racial differences and how that affects TNF inhibitor usage and sustainability.
Thank you, Dr. Borg. So I found this very interesting study. It's abstract number 1780. So they studied the racial differences in psoriatic arthritis patients in a very large US real-world database. I think the downside to the study is that they based it on the computer diagnosis, but either way, they have a very high number of patients. They had about 26,000 patients who were included. And out of this, some of the interesting points that I saw was 95% of this were Caucasians and 5% were African Americans. But it's because of the population being this much, it's still a good number of representation. And they wanted to find what were the differences in the TNFIs, the NSAIDs, or the DMRs that was used. And among the tumor necrosis factor inhibitors, about 51% of the Caucasians used TNFI and 41% of the African Americans. And even though I understand that the percentage might not be different, but when they did the probability scoring, there was a significant difference in the usage of TNFI.
And I think this goes along the lines of many, many abstracts showing racial disparity, and it has to be more than downright access or lack thereof, because if you actually get prescribed a drug that's an advanced therapy, you've had some degree of access to a touch point with the healthcare system. And even to make the diagnosis, the code of 696 or 721, however they did their coding to identify psoriatic arthritis, somebody coded it, so they've had access. So it just seems that we saw a lot of it in lupus. We saw racial disparity amongst every single study, JIA, each disease that looked at it at this meeting. And I think we have to have our eyes open. And as one of the speakers said in one of the sessions, is that if you're colorblind, you're lying. She didn't quite say it like that, but she said we all see color, race, socioeconomic status, and it's our unconscious bias. And hopefully after this meeting, we'll go home and say back to our practices and say, I really have to make sure that I, pinch myself now and then, that I'm giving the best treatment for every patient walking in, not just the most literate, most demanding patients who are, say, like me, right? So patients, all of them, it's really important that we actually give access where appropriate and have more level playing field on outcomes such as lupus nephritis or JIA, rehospitalization, et cetera, et cetera. So I think this meeting really tried to give a spotlight to that. So thank you for presenting that. So we hope everybody enjoyed their room now. There will be loads and loads of information to come. So please keep following us, and we all tweet away on a regular basis. Thank you.